Today's topic is the complement system. Now the complement, we know it is found in the sera, that means the blood plasma of all the mammals and many other animals including the birds, amphibians and the fishes. Second, the complement, it is non-specific reactivity. It shows non-specific reactivity. The complement, it's a non-specific serological reagent. That means that the complement from one species, it can react with the antibodies from the other species. The third is the complementary concentration. The complement it constitute about 5% of the normal serum protein. So the complement, it constitute how much? 5% of the normal serum protein. So its concentration does not increase as a result of immunization. Complement, it shows temperature sensitivity. It is heat labile. What does that mean? It means that it is sensitive to heat. It shows cell cytolytic activity. That means it shows cell destroying activities. So it can spontaneously denature at room temperature, and it is completely destroyed within 30 minutes at 56 degrees Celsius. The serum that has lost its complement activity due to heating, it is said to be inactivated. Complement does not bind to free antigen or antibodies, but only to the antibodies that have combined with their antigen. So it shows binding specific. So this binding, it is necessary for activation of classical complement pathway. So remember, IgG class that fix complement. Not all the class of immunoglobulins can fix a complement. Only IgM, IgG1, IgG2 and IgG3 can fix complement, whereas IgG4, IgA, IgD and IgE cannot. So the site of complement binding, it is located on the Fc piece of the Ig molecule. Specifically the CH2 domain on the IgG and CH4 domain on IgM. The other is the fixation of complement. It is not influenced by the nature of antigen, but it is determined solely by the class of the immunoglobulin that is present. Now let us discuss the complement components. The complement system, it consists of complex network of at least 30 distinct serum protein. So these protein includes not only the complement components but also the properdin system proteins and control proteins. The complement system was initially associated with innate immunity, which provide a rapid and non-specific defense against the pathogen. However, it has been recognized that the complement protein, it also play a role in acquired immunity. Now the complement, it is composed of set of distinct protein fraction that is labeled from C1 to C9. So these fractions, they are involved in cascading series of reaction that leads to immune responses. Remember the C1, it exists in serum as a calcium ion dependent complex, which can be separated into three proteins, that is C1q, C1r and C1s. So this means that the complement, it is made up of total of 11 different proteins. The fractions are named C1 to C9 in the sequence of cascading reaction, with C4 occurring after C1 and before. Now when the complement component, they binds to erythrocytes antibody complex, it is known as EA. So when the specific complement component attached to this complex, the result is erythrocyte antibody complex. The number indicates that which component are involved, like EAC 14235 would suggest that the component 1, 4, 2, 3 and 5 have reacted with the complex. So if a complement component become enzymatically active, a bar it is placed over its number, like C bar. So it is also represented as C1. The fragments that are cleaved from the complement component during the cascade they are indicated by small letters like C3A and C3B. They are the two fragments that are derived from C3. Inactivation of the complement components are indicated by prefix I. So like C3b would be an inactivated form of C3b. Now complement, it is normally present in an inactive form in the body. However, it can be induced into the active state by various stimuli, including the binding of antibody to the antigen. So when activated, the complement components undergo a series of reactions in specific sequence that is known as cascade. So these in the cascade, each component act as an enzyme, leading to cleavage of components into dissimilar fragments. The larger fragments usually continue in the cascade, while the smaller fragments are relieved and often have some biological effects. Now, what are those biological effects of the complement system? There could be lysis of cell and bacteria, so that helps to destroy pathogens, promoting virus neutralization, preventing viral infection. Opsonization, it is a process that enhances phagocytosis, that is the engulfing and destroying of the particular antigen by the immune cells. So immune clearance, that removes the immune complexes from circulation and deposit them in spleen and liver for further processing. Amplifying the inflammatory process leads to increased vascular permeability, the smooth muscle contraction and release of the histamine. From the mast cells. So this inflammatory response, it is essential part of body defense against the infection and injuries. The complement activation, it can occur through 3 parallel but independent pathways, each different in its initial step. So once the activation of the C3, a key component of the complement system, takes place, the subsequent steps in the cascade are common to all pathways. So these 3 pathways, they are the classical pathway, alternative pathway and lecithin pathway. The classical complement pathway, it is the specific sequence of events that leads to complement activation. The classical pathway, it is initiated by binding of complement protein C1. The classical pathway involves the binding of protein that is C1 to the antigen antibody complex. So this complex, it is traditionally represented as EA. The E stands for the erythrocytes and A stands for the antibody. So where the recognition unit of C1 is the C1q, which binds to the Fc region of the immunoglobulin IgM or IgG. So once C1q it is attached, it activates the C1 esterase, an enzyme. C1 esterase has ability to cleave multiple molecules of the C4. So C4, it is a part of the complement system that is split into two fragments, that is C4a and C4b. The C4b, it binds to the cell membrane along with the C1 to form a complex. In presence of magnesium ions, the C4b, it cleaves C2 into C2a and C2b. So it has a kinase activity, the C2b, it has a kinase activity and increase the vascular permeability. The C4b2a complex that is formed, it has a enzymic activity and it is referred as classical pathway or C3 convertase. So the C3 convertase, it cleaves C3 into two fragments, C3a and C3b. C3a, remember, it is an anaphylactic toxin that is released in medium. And C3b, that remains cell bound along with C4b2a to form a trimolecular complex that is known as C4b2a3b, that is known as a C5 convertase. So the membrane attack phase begins with the C5 convertase, cleaving C5 into again two fragments, C5a and C5b. The cascade continues with the C6, C7 and joining together to form a complex that is C567. So part of this complex binds to cell membrane and prepare it for lysis by subsequent complement components, that is a C8 and C9. C8 and C9, it joins the reaction and leads to formation of holes on the cell membrane. So these holes disrupt the osmotic integrity of the membrane, leading to release of the cell contents and ultimately leads to cell death. The alternate pathway, it is activation of a C3, which is a major component of the complement system. In the classical pathway, the C3 activation, it is achieved by formation of C4b2a, that is C3 convertase. So in the alternate pathway, C3, it is activated directly. So the first step in the alternate pathway involves binding of C3b to an activator. C3b, it is continuously generated in the small quantities in the circulation where it exists in a free state, and it is rapidly inactivated by serum protein factor H and I. When C3b binds to an activator, it is protected from inactivation. Bound C3b, it interacts with the serum protein factor, that is factor B, that is also known as a C3 proactivator. So this interaction leads to formation of magnesium dependent complex, that is C3b. So this complex, it is cleaved by another serum protein factor, that is factor D. So this cleavage results in production of two fragments, BA and BB. So fragment BA, it is relieved in the surrounding media, while this fragment, it is remained bound to C3b. So remaining fragments binds to C3b, creating a esterase complex that is known as C3 convertase, that has an enzymatic activity. The function of properdin, also known as factor P, it is to stabilize the C3 convertase. That stabilization is important for continued hydrolysis of C3 and initiation of further steps in the complement cascade, similar to the classical pathway. The third pathway is the lecithin pathway, in which the complement system can be activated. So lecithin pathway, with the primary lecithin involved is the mannose binding lecithin, present on the surfaces of microorganisms like strain of Salmonella, Neisseria, Listeria, fungus like Cryptococcus and Candida albicans. So when this mannose binding lecithin, it binds to foreign surface of pathogen specific proteases known as MASP1 and MASP2 binds to MBL. So this complex MBL here, the MBL MASP complex, it acts as an initiator, analogous to CA1Q in the where the initiator was C1Q in the classic pathway. So once this complex is formed, it causes cleavage and activation of C4 and C2 complement proteins, that results in formation of C4b2A complex that act as C3 convertase. So this activation occurs without the need for specific antibody binding. So the lecithin complement pathway, along with the classical and alternate pathway, converges at the membrane attack protein. Eventually, so this MAC complex contains a complementary protein C5b, C6, C7, C8, C9. So the formation of MAC complex, membrane attack complex, that leads to creates a channel through the target cell's membrane, allowing small molecules to fuse freely across it. So that would disrupt the osmotic balance of the cell, leading to water entry and loss of electrolyte. So as a result, the target cell cannot maintain its osmotic stability. That is why it is ultimately killed. Now let us discuss the inhibitors of the system. They can be C1 esterase inhibitor, other is S protein, so they are the inhibitors. The inactivators are factor 1, factor H factor, and anaphylatoxin inactivator. Now, what are the syndromes that are associated with genetic deficiency of complement components? The C1 inhibitor, the syndrome is hereditary angioneurotic edema. Early component of classical pathway, the syndrome is SLE and other collagen vascular diseases. C3 and its regulatory protein, the syndrome associated is recurrent pyogenic infection. C5 to C8, that could lead to bacteremia, mainly with gram-negative diplococci. So these are the syndromes that are associated with the genetic deficiency of the complement components.